We spend a lot of time on the phospholipid membrane, the bilipid membrane, because in terms of our class, it is one of the primary ways that physiological functions actually occur. Allowing something to cross through a bilipid membrane would, for example, allow the cell to suddenly have access to something it didn't have access to earlier, or would allow the cell to produce a product that it hadn't been allowed to release earlier. And so the plasma membrane, in terms of physiology, is probably the most important structure of the cell, at least in terms of what we study in our class. The bilipid membrane, or some sort of derivation of it, exists in all of our cells, plus many membranous structures, for example, the organelles inside of our cells. And this is true of all animals that I know of, all plants that I know of. Bacteria even have a bilipid membrane such as this. So it has proven to be a conservative structure across all living things and is incredibly important. This semi-permeable membrane allowing only things through that you really want through in that moment is incredibly important to physiology and to life as we know it. So we will be discussing cell structure in an abbreviated way, but the bulk of what we're discussing today is going to be based on the phospholipid membrane, the bilipid membrane that comprises the external part of most of the cells of our body. And basically, the purpose of the bilipid membrane is to keep most everything out or in, except those things that you want to leave the cell or to enter the cell. So the atoms that we're going to be working with here are carbon, hydrogen, oxygen, and phosphorus. And nitrogen actually won't be listed here, but sometimes is a part of this, and I'll bring that up when it comes about. The first thing we're going to be drawing is what we call a glycerol backbone. And it's 3 carbons. They are surrounded, as always, carbons always have 4 bonds. As you can see, the central carbons there, the 3 carbons in a row, are bonded to hydrogens and oxygens, or to each other. And so we label this as glycerol. So we have 3 carbons attached to oxygen, and then you fill in around there with hydrogen. Now I'm going to switch colors because the rest of this structure is going to be considered a kind of a separate functional group that is attached to the glycerol. So glycerol is comprised of these 3 carbons with these oxygens, ester bond, my favorite bond, just because it has a cool name, right? And what we're going to do is we're going to make fatty acid tails, hydrocarbon tails. And just like their name implies, it's carbons with hydrogens. And they're going to be even numbers of carbons, starting with the one up at the top that is double bonded to oxygen. And so the first one I'm going to make is going to be 8 carbons long, and I'm just filling in all the hydrogens. There's always going to be hydrogens around there. The second one I'm going to make, the middle one is going to be a little bit different. I'm going to put a double bond in there. As you can see, the first one only has single bonds. In this one, I'm going to put together 1 double bond as part of it. There's the double bond. And so what happens when you have a double bond is that the actual configuration of the hydrocarbon starts to kink and kind of turn off in a different direction. I'm not great at drawing this stuff, but you get the idea. Now, because those carbons have a double bond, they will receive only 1 hydrogen. The other ones all have 2, as well as the bond that they have to each, down the length of the hydrocarbon. And the last one, we'll just go ahead and make it a little bit shorter. These hydrocarbon tails are usually going to be longer, like 14, 16 carbons long, or even longer than that, but they can be of varying lengths. I couldn't draw out so many carbons just because I'd run out of space on the paper. All right, so these are fatty acid tails. And 2 of them are what we call saturated fatty acid, which means that they have all the bonds possible associated with hydrogen. But when you put a double bond in that middle one, that is considered an unsaturated fatty acid tail because it does not have all the hydrogens possible. It's possible to break one of those double bonds and add more hydrogens, and that's what makes it either saturated or unsaturated. So as you can see, you can have variability there. You can have some saturated and some unsaturated, and this is very typical. So we have glycerol as a backbone, and then we have these 3 fatty acid tails that come off of it. And this is the way that we actually abbreviate it. This would be a typical way that we draw a glycerol backbone. And then what we do is instead of drawing all those C's and H's, we kind of know that there's a lot of carbons there. We know that it's a fatty acid tail. We know that's what it's made of. We know it's made of carbons and hydrogens. And so we simply use the intersections of angles to demonstrate the presence of a carbon. So there we have we infer that there's a carbon there, even though right next to bonded with that double bond to the oxygen, even though we didn't actually draw the C and the H and all those things, we know that it's there. So every time you see a point on one of these jagged lines here, every time you see a point, there's going to be a carbon. And so I'm doing my easiest ones first. I'm doing the saturated ones first. Now I screwed up the unsaturated one, but here it goes. There's our double bond. We are inferring there are carbons everywhere along the line. We all know how many hydrogens would be bonded to them. And I think what I screwed up was that I drew too many carbons. But anyway, you get the idea. So we have this kink. The center one has the double bond, so it has this kink. And it actually, unsaturated is helpful because when you have fully saturated fatty acid tails, they tend to lie together really easily and they become a solid very easily. And this is where when you have saturated fatty acids, when we discuss this in nutritional circles, what we're discussing is something that can easily clog up an artery, just kind of lie against the wall of an artery. So saturated versus unsaturated. Unsaturated is better in that way. It remains more liquid. Those kinks help it to not bond and stick so easily. All right, so the entire thing is called triglyceride. Now the triglyceride is a name for basically blood-borne fats. The fats that circulate through your blood are in the form of triglyceride, and so when you get a CBC taken, when you need a complete blood count taken, generally speaking, they're going to look for triglyceride levels. A lot of triglycerides are stored, for example, in your liver or in different parts of your body, but some of them circulate through your blood as well, especially if you have excess numbers of them. So triglycerides are a good number. The triglyceride number in your blood is a good way to indicate whether you have too many fats circulating through your body, basically. And then triglycerides are also the primary form in which we take food in, the way that we take fats into our body. Now the next structure is going to be drawn in somewhat of a comparison with triglycerides. We're going to be actually drawing a phospholipid which has a very similar makeup to a triglyceride. So first of all, we draw our glycerol background. I'm not going to label it. You should recognize it by now. CH2, CH2. Here we go with our hydrocarbons with that double bonded oxygen at the top, at the tippy top. And I'm going to go ahead and draw our second one, this time with the right number of carbons, I think, based on the sketch that I did previously. There's our double bond. So we have saturated fatty acid, we have an unsaturated fatty acid, and instead of linking a fatty acid to that third carbon, we're going to put something else there. We're going to put a phosphate group there. Switched colors to blue because you need to be able to recognize this is a third thing attached to this molecule. I kind of had to squeeze it in at the top there, but it's basically a P, a phosphorus with 4 oxygens surrounding it. There is a double bond which we assign to one of the oxygens, but generally it would be equally shared basically amongst them. You see a negative charge there, which is the reality is that if an oxygen only has one single bond, then what is open is basically an unpaired electron, and that allows you to have that negative charge. So phosphate groups, generally speaking, are negatively charged, depending on what they're bonded to, of course, but we usually think of it as having a negative charge. And then that oxygen at the end there is going to have an R group. Now the R group sometimes has nitrogens as part of it and sometimes does not. I thought maybe the details of the R group was not quite as important with this particular lesson. What you do have to do is be able to recognize a phosphate head, P with 4 oxygens around it. And it's oftentimes denoted simply with a P with a circle. It's an incredibly important structure, an incredibly important molecule that we'll see come up again and again. Now you'll see that I labeled the phosphate head and I used the word polar which is the same thing as saying hydrophilic. And I went ahead and labeled the fatty acid tails again, which I will now use the word nonpolar, which means hydrophobic, and that's going to be coming up very shortly. So the entire structure is called a phospholipid, 2 fatty acid tails, a glycerol backbone, and a phosphate head. The phosphate being polar, the fatty acid tails being nonpolar. It means that they don't really like being around each other, so they kind of twist their way away from each other. So let's move on to a generalized discussion of the cells of the body. Here I'm going to be drawing sort of a three-dimensional view. I hope I'm doing it very well. A three-dimensional view of a cell. Usually we picture cells being, circles, but I'll just draw it in an oval, kind of give you the idea we're looking at the edge on. And the reason I used the bright orange color to indicate fatty acids is because I really didn't want you to miss them. The bulk of a plasma membrane is comprised of those fatty acid tails, and so I've used that orange color here as well. And this time only, I'm going to go ahead and draw phosphate heads on there to indicate that you should keep in mind that phosphate heads are going to be part of this. We never draw anything like this. We think of a bilipid membrane, we think about the plasma membrane as being made of lipids, being made of fatty acid tails, being made of fats, non-polar structures. It is worth a couple times considering that there is also that phosphate head on there, which is in fact polar, making it possible for the plasma membrane to actually be present in the fluids of the body. All right, I went ahead and drew the nucleus, and I'm also going to make a little play at drawing some organelles, just to keep in mind there's organelles there. They're going to be coming up as part of a couple of lessons we're going to be discussing in the future. So smooth endoplasmic reticulum, rough endoplasmic reticulum, you got some mitochondria there, got some ribosomes, little lysosomes. There's a bunch of structures here. And I'm just going to be generally calling them for now organelles, tiny little organs inside of the cells of the body, just so you remember that they're there. But the main structure I want you to keep in mind of this cell is going to be the nucleus, because the nucleus is the structure that is most visible in the future when we discuss histology, and we're going to be discussing different types of cells. It's helpful just to keep in mind, yes, it's a cell because, boy, I see the nucleus right there. So oftentimes when we think of cells, we might be thinking of, for example, white blood cells circulating through our blood, or what I'm drawing here, an epithelial cell sheet. And I'm going to go ahead and draw the nuclei inside of there so that you can kind of keep it in mind. You can, kind of look at the cell structure. You can kind of see the plasma membrane there, and you can almost always see the nucleus inside of a cell. But I also wanted to draw some of the more unusual cells of the body. Here's the neuron. It's got dendrites, cell body, axon. Sometimes students need a little bit of a reminder that this is in fact a cell, and so I usually try to draw it with a nucleus. Just so you keep in mind, yes, it has nucleus, and yes, it has organelles inside of that cell body as well, but they're not portrayed here. You usually won't see them under a light microscope, but you probably will see a nucleus actually. Here is kind of a spindle-shaped cell, a smooth muscle cell of the body, and the orange around the outside indicates the plasma membrane, and the black circle in the middle is the nucleus. And here are some skeletal muscle cells, which are incredibly long, and they are what we call multinucleate, which means that each of the cells actually has multiple nuclei, indicating that over evolutionary time, there were individual chunky little cells that kind of melded together into longer ones, and they retained their nuclei. So look for the plasma membrane, look for the nuclei, and you can be assured that these things are in fact cells. So here's my last box, just my little introductory lesson. And now what I'm going to do is I'm going to take a chunk of that hypothetical cell and we're going to go ahead and blow it up so we're taking a closer look at that structure. When we draw the plasma membrane, it's worth considering in the first place that it is a fluid mosaic. The fluid mosaic model is the model with which we engage with the plasma membrane, getting the idea that it's not one solid structure, but it's actually kind of a sea of things floating around each other. Here, I'm actually going to start us off drawing some proteins. The one that I drew up above there is going to be a protein floating outside of the cell. These ones that I'm drawing here are going to be embedded in the cell membrane. So since I know where I'm going with my plasma membrane, I can draw the proteins ahead of time. You may not want to do that. I just find it's helpful. Then you don't have to like draw over your plasma membrane. But you can start drawing your plasma membrane. So the 4 in the middle there, they're all embedded in the membrane, and then there's one inside of the cell. Now what I'm going to do is I'm going to fill in our phosphate groups. Remember that we used blue for the phosphates. Take a look at our phospholipid. We used the blue color, so I'm going to go ahead and draw a continuous barrier of phosphate groups all the way along the length of the plasma membrane, basically only making way for the proteins. And then there's going to be other structures in there like cholesterol and some carbohydrate structures and things like that. But basically, you're going to see this fairly, this fairly continuous line of phosphate groups because they are polar and because they can endure the presence of fluids, of polar fluids, such as the fluids of your body and the fluids of your cells. Now, on each of them, I'm going to draw 2 fatty acid tails. Again, take a look at our phospholipid that we drew up ahead. Each of that, I know that I'm not drawing the glycerol backbone here, but we don't really have to draw that. We've already drawn it in detail. We've inferred that it's there. You know that it's there. The most important thing we draw in this model is going to be the phosphate heads and the 2 fatty acid tails, which tend to kind of curve around each other because a lot of them are going to be unsaturated and double bonded and so on. So you'll see lots of wavy lines there, which means they don't like just fit against each other and sit solid. They tend to move around quite a bit. Remember, a fluid mosaic. And the cholesterol embedded in there is going to help with structure and to keep things moving around, although I'm not drawing any cholesterol structures here. Thought this would be the time we'd go ahead and label our phosphate heads. You'll notice that they are on both sides, a bilipid membrane. There are phosphate heads. I'm only labeling the ones out here, but keep in mind that if you go to the other side of the membrane, there's also phosphate heads. They are also polar, also called hydrophilic or water-loving. And you know what I'm drawing next? I'm going to be labeling next the fatty acid tails, which are the opposite. The fatty acid tails are nonpolar, hydrophobic. They do not enjoy, it's basically kind of impossible for them to exist in a polar environment such as the fluids of the body or the fluids of the cells. And so they kind of depend on the phosphate heads to do that job for them. And then they're able to form this nice lipid fatty membrane that keeps lots and lots of things out of the cells or inside of the cells. Now, traditionally, when we draw a membrane side to side like this, the outside of the cell is up above, and so we call this the extracellular fluid, also labeled, also abbreviated as ECF, but most commonly actually called IF, or interstitial fluid, the fluids that circulate around the tissues of your body. And the inside of the cell is going to be portrayed down below, so we call it intracellular fluid, or ICF abbreviated, or it's also called the cytosol, which is that jelly-like fluid inside of your cells. So now we can go ahead and label this chunky little protein as one of the external proteins, this one here as one of the internal proteins. Oftentimes the proteins we're most interested in, though, are the ones that are actually embedded in the membrane. And especially these long ones that I've drawn here, which we'd call transmembrane proteins. They actually traverse all the way from the inside, you see, to the outside, or from the outside to the inside. And they're embedded in the membrane. We're going to take a closer look at them I think in the next video. So we have another transmembrane protein here. And here we have a protein which is embedded in the membrane but faces the inside of the cell. And here we have a protein which is embedded in the membrane but faces the outside of the cell.